Hello and welcome to the video which I will explain an introduction to scientific writing and the formal laboratory report requirements and guidelines for immunology laboratory MLS 326. If you look here, we are on the home page of the course website. All students, regardless of which laboratory section you are enrolled in, have access to the same website. So we only upload things on one website. The only difference will be due dates, and there are 2 different course syllabi posted this year. There'll be syllabi for each lab section. They are the same except for the schedule and the instructor's contact information. So again, this is the welcome landing page. And we are going to go to Introduction to Scientific Writing, Formal Lab Report Requirements and Guidelines. You can click on the folder, the content folder on the course homepage, or the course menu is along the left margin. You can also access that same content folder there. So I'll click on it from there. This video will be posted right here. You'll be able to watch this after I record it as often as you'd like. And the ICMJE citation and reference format is the format that we use for all MLS courses in this department. And you should be familiar with that format. And here is an overview of a scientific literacy workshop from MLS 122 Genetics Lab and another presentation that you may or may not have participated in the recent years. And then there's some information about how you can avoid plagiarism. Okay? Intentionally or unintentionally, plagiarism is not accepted. So let's click on here, the reference and citation format you can look at. You've seen it before and you can click on there. Okay. So that brings us to this document. I highly recommend that you print this out and you keep it by your side when you are preparing your formal laboratory reports. You will not prepare a formal written laboratory report for every lab exercise. There will be a certain number. Please refer to the course syllabus under laboratory reports for the number and the groups. There are different groups and students have to choose one from each group. Okay? If it is a week where you are performing more than one assay, please check with your instructor before you leave the laboratory that afternoon or morning and ask them which of the 2 they would prefer you to write the formal lab report for. Sometimes there's a preference based on your results or your patient information. Okay? So again, please refer to the course syllabus for which labs you are eligible to write up a formal lab report and how many you must write up. The other labs, you will be submitting your data and some responses to discussion questions. So general considerations, you can read this here. Okay. Make sure that you use spell check. Spell check does not always catch every scientific word, so you'll have to manually check for spelling and grammar. And also please be aware that your first draft should never be your final submission. Okay. You typically have 1 week, unless otherwise noted, to complete the formal written lab report. So please use that time wisely. Don't wait till the last minute. These will require a substantial amount of writing and reading to prepare to write the lab report. Reading of textbooks, journal articles, and searching for some information to cite in these lab reports. Also, if you would like to meet with your instructor, please don't wait till the day before. All formal lab reports will be submitted through SafeAssign. It's a plagiarism check software that the university uses. Please submit at least 24 hours before it's due so you can check and see what percentage it flags. There is no percentage number, but if you're at a very high percent, it probably means that you didn't do a good enough job in paraphrasing your cited information. Okay, so you want to give yourself ample time to correct some of that and resubmit. Lab reports cannot be emailed, so they are to be submitted through SafeAssign, and then they will be printed out and brought to the laboratory on the due date, a hard copy. Lab report format, double space using Times New Roman font, 11-point size font. Please use the size in the font and the spacing that is listed here. Okay. There are 5 sections that you will be including. They're in this document. Please use the headings that are indicated in this document and bold them or indicate that they are a heading. Please also have the lab report written in the order in which the headings appear in this document here. The minimum requirements you'll see here, listed 1 through 6, the original patient requisition that you must sign and date on the day you perform the assays, that will be given to you in lab. You're going to need that for the patient information about the assay you're performing, information about the specimen type, also current diagnosis in any medical history, including previous diagnoses, including pregnancy history if applicable, and or transfusion and transplantation history. So you will need that information to help you write this. That must be submitted. Your original data or results, that must be signed by your instructor before you leave lab. We do not want you typing up your data. We want the original data. Okay. In-text citations must be used. Please refer to the ICMJE. We use superscript numbers, not author name and date. And those superscript numbers need to go at the end of every cited sentence, not at the end of an entire paragraph and not in the middle of a sentence. Your literature cited page is another words for reference page. We use the words literature cited. That is a separate page at the end of your lab report where you will include all the references that you used. If you cite something in the body of your work, it must be included on that literature cited page. We want the entire first page of any journal article that you cite. We don't want just the abstract, we want the entire first page, and if you have annotated it in any way, please include those annotations. And again, must be submitted through SafeAssign and then a hard copy brought to lab the date it's due. Okay. If the minimum number and appropriate required references are not included, which are listed at the end of this document, the lab report will not be evaluated by your instructor and you will not receive credit, and a grade of 0 will be recorded. There has to be citations and references. Okay? You do not get to redo your lab reports. So please follow these guidelines. If you don't do well on a lab report, you don't get to redo an extra lab report. So take your time. These are a work in progress. You will get ample feedback from your instructors. So please read our feedback. We spend a lot of time evaluating these and providing feedback. These are not easy, and this is the first time that you've done, I think, in the MLS department, this full formal lab report. I believe that you've done bits and pieces or different sections that will be included here, but not the full lab report. The sections are here. It's based on 50 points, but that's converted to a percentage. Okay, so the sections are 10 points each, and then if you've got a 40 out of 50, that would be an 80%, a 46 out of 50 would be a 92%, and so on. Okay. You are presenting in the introduction, you are presenting this piece of work to a reasonably knowledgeable reader, meaning they have some understanding of science. You don't have to go over basic science concepts. In the introduction, I'm going to use, for the sake of this example, an indirect ELISA assay in which we would be testing for the presence or absence of antibodies in a patient plasma or serum sample against HIV, human immunodeficiency virus that can cause AIDS in humans. So if this was the case, in the introduction, I would want to introduce the assay, what type of assay. I'm running a non-competitive, heterogeneous, indirect ELISA, which stands for enzyme-linked immunosorbent assay. And what am I trying to detect? What's the clinical significance? Why am I running this? I'm running this to detect, like I said, antibodies directed against HIV. Okay. Why might that be important? Who would get tested? Potentially who would get tested in general, who might we test? Okay. And what kind of, so we're talking testing about antibodies directed against HIV, and again, what kind of disease does HIV cause? Okay, and a little bit of information about the disease, the disorder, and why the testing is needed. Why would a physician order this test? This section should also include specimen requirements. So for this case, you would be drawing a whole blood and you would be using the plasma or serum. Okay. If there's any special handling or storage, you need to put that, how long the specimen is good for once it's drawn. Do you have to freeze it? Does it have to be tested at room temperature? Okay. So that's that section. That's a good section to maybe use a journal article to cite. Now, when you are citing any information, you want to write it in your own words, but you still have to cite it. Information from lab lecture, you do not have to cite. That is considered general knowledge. You cannot cite your laboratory instructor or the laboratory procedure. That information we got from primary sources. So you need to cite a textbook, a journal article, something like that. Okay. Any citation that you have needs to be followed up with an explanation as best as you can in your own words. The point is that we want to get a sense of, do you understand what you're writing? And please consider where you put cited information and how it's incorporated. Don't just stick random sentences in your introduction or methodology or discussion. It needs to be well incorporated. So you will get the citations and the references will be evaluated on, are they the proper number? Are they the appropriate source, types of sources, but also how well they are incorporated. Okay? That's going to take significant amount of reading before you can write the lab report. And maybe you might have some questions for us. Methodology. This is not a step-by-step procedure. That's probably the biggest mistake that students make when they're inexperienced at writing these. This is why we want you to read some journal articles, because the journal articles are set up like this, some kind of introduction or abstract, a methodology section. So for example, if I'm performing a non-competitive, heterogeneous, indirect ELISA to test for the presence or absence of antibodies directed against HIV, I want to restate that in the methodology, and then I want to describe what that is. So a couple of things in the methodology: the type of the test, the name of the test. If it's an acronym, you need to tell us the entire test name. Okay. If you've already done that in the introduction, then you can just use the acronym, such as ELISA, in the methodology. You need to always identify the source of antibody or antibodies, if there's more than one, and antigens. Okay, immunology, always be thinking antigen, antibody. Okay, so did it come from the patient plasma? Did it come from a nasal swab, an antigen? Is the antigen, in this case, the antigen is a reagent in the kit. But in this case, in the indirect ELISA, it's called an indirect ELISA because there's two antibodies. The primary antibody, the source of that antibody in this indirect ELISA would be the patient's plasma sample, if it's present. The antigen is from the kit. There's a secondary antibody that's an anti-human antibody that will bind only if the primary antibody in the patient's specimen has bound to the antigen. So again, here, there's a primary antibody from this human specimen, the patient, and a secondary antibody that's provided in the kit. You want to tell any specifics if you know. In this case, the antigen would be a simulated and HIV antigen. Again, the secondary antibody is a reagent. It's an anti-human antibody. And in an indirect ELISA, the concept is based on being able to visualize this. So there's an enzyme linked. You want to list what the enzyme is. There's a specific enzyme used here. It's horseradish peroxidase in this case. There's also a substrate that gets cleaved by the enzyme. It's a colorimetric substrate, meaning that it has the ability to change color. And it does that if everything's bound together. And the substrate here is TMB. It stands for a very long chemical name. In this instance, I might also add an illustration, a labeled illustration, showing what binds to what. If I wasn't going to label an illustration showing that, I have to say that out in words. Okay? Again, not a step-by-step procedure, but an explanation of the basic test methodology, the immunologic principles of the assay. But you still want to consider the what, the how, and the why. So going back to my example of my indirect ELISA for the detection of antibodies directed against HIV, I do want to put all the components, how they're used, what binds to what happens if there are antibodies, what kind of reaction occurs when there's not. I want to include any incubations, how long I incubate, what temperature, that's important. Okay? And in your cold agglutinin titer assay, you're incubating in the refrigerator. Why? Okay, that tells us that you understand what's going on immunologically in this assay. Okay, so times of incubation, temperatures of incubation, and why. So the what, the how, the why. Okay, and then always include any quality control. So in this case, we perform a positive control and a negative control. Okay, and what's in those controls. The other thing in this particular assay, everything is run in triplicate, ×3. That has to do with the fact that we use microtiter wells, and they're very, very tiny. So when you're pipetting, you can have spillover, and there could be contamination. So in this case, to get accurate results, or results that you can report, valid and accurate, your QC has to work as properly. What do I mean by QC works? It has to yield the expected outcomes, and 2 of the 3 wells have to have identical results. Okay, so I would include all of that. The data and results is pretty easy. There's some standard deductions. So if you have a figure illustration, there's no figure legend, there's no labels on an illustration, that's -3 points. And if you don't get accurate results, that's an automatic 5-point deduction. So you might want to look at what your options are for eligibility for formal lab reports at the start of the semester. Look at your schedule, but then also be flexible. If you thought you were going to submit a lab report on such and such lab and you didn't get good results, you might want to reconsider that if there's still options remaining. Okay. So this section, again, your original results and any kind of general explanation you might have to say about those results. Okay. Discussion. This section, there are 5 items that you need to address. Okay. This is the other more difficult section because it is requiring students to correlate a lot of information, which hasn't had to be done at this level before. So you want to provide a clear, concise summary and interpretation of your findings. Okay? That's pretty straightforward. Okay? Not just saying it was positive, but in the indirect ELISA, the wells turn blue. That means it's positive. That means that there are antibodies directed against HIV in that sample or the positive control. Okay? Provide an evaluation of the quality control. Okay? Did your quality control perform as expected? And what does that mean about your test validity? Provide a discussion of probable experimental error. Here you want to write something that is plausible for your experiment. So for me, with the ELISA, it might be pipetting in that I cross-contaminated, or there's several washing steps. Okay, if I didn't wash properly, or I left wash solution in it, or I skipped a washing step. But you can't just say human error, and don't just say pipetting error. Describe the specific error and make sure it's something that could have happened in your experiment that you performed, not the experiment in general. Okay, and I only need one good one. The fourth point of talking here or discussion is correlate your result with a patient diagnosis, current diagnosis, medical history, and transfusion history. Medical history may include a pregnancy history if applicable. Okay, provide a scientific explanation. So here you now need to look at all the information provided on your patient requisition. This is going to be different than people sitting at your lab bench. Students are going to get different patients with different medical histories, different diagnoses. So now we're asking you to put the pieces together, not as in the introduction where you're just generally talking about the assay and why it's relevant, but here, what does it mean to the health status of your patient? Okay, so if I detected antibodies against HIV in my patient's plasma, that means that my patient has had an immune response. They were exposed, they contracted HIV, and they have antibodies directed against it. Okay. And the last discussion point here is an interpretation of the clinical significance. Now we're not talking again general clinical significance as you did in the introduction, but clinical significance as it relates to your specific patient. So what might that mean in my case? If they've been exposed and they've developed antibodies, if they're not already on treatment, we might want to think about that. We might want to think about, are there any additional tests? Are you performing a screening assay that will then require a different assay to confirm something? Okay, so each of those needs to be addressed. There'll be a maximum of 2 points. 2 times 5 is 10. And then your literature cited section is 10 points here. Please read through this carefully. You need a minimum of 3 references of 2 different types. 1 journal article must be used, and it must be a journal article that was published within the last 10 years. You are certainly able to use older articles for historic reference, but they have to be in addition to the 3 sources that you're using. So by all means, you can include more than 3 references, but a minimum of 3. Okay. Websites, you cannot just cite a website. If you go on the FDA website and you access an article, you must cite the article, or you must cite a specific location for the website, and please look at the ICMJE format for citing that. Okay, but you can't just cite an entire website. There's a plethora of information. If you're citing your textbook, which I strongly encourage you, once you cite it once and it's in the correct format, save that to a Word document. You must cite the pages that you've used in the textbook, not just an entire textbook. Okay? If you have any questions, please see your laboratory instructors for extra help, and happy writing and learning.